Hey, welcome to the series of Being a Microbiologist presented to you by MindNotTired, where we discuss about some very basic concept which a microbiologist mind must know. So in this video, let's discuss about mycology or the study of fungi. So by the end of this video, you should be able to describe the structure of fungus, classify the fungus on the basis of various bases, lab diagnosis of fungal infection, and at last we'll be summarizing the whole video with some viva voce questions. So first pertinent question is, why should we study mycology at first hand? So there are various reasons. First of all, in order to develop new antifungal drugs against the uh, existing fungal infections. Secondly, antifungal susceptibility testing. This is done in order to provide effective and personalized treatment to the person suffering from the fungal infection. Next, there are many fungi which are becoming resistant to antifungal drugs, so we have to know about them. And lastly, there are emerging new fungal infection day by day. This is due to change in host or virulence factor. So properties and structure of fungus. So we all have read about fungi in some or the other classes in our school days. So it is ubiquitous, means it is found in every place of this world. It is a eukaryote. It has got rigid cell wall consisting of chitin, mannans, and other polysaccharides. So as you can see in this diagram, we can see this yellow colored chitin, then beta glucans, and then manoproteins. Now this cell wall structure is also of clinical importance, which we will be seeing in further slides. The plasma membrane of the fungi consists of sterols, especially ergosterol and cymosterol. And lastly, they have got heterotrophic mode of nutrition, means they can be saprophytes or parasites. Now, classification of fungi. We've got various bases to classify. So first is the based upon morphology, based upon spores, based upon vegetative state, and based upon location. Based upon morphology, so fungi can be divided into yeast-like, molds, and dimorphic. Yeast are the fungi which undergo the asexual mode of reproduction known as budding, and here the budding is complete, means the daughter cell separates out from the parent cell and they form two separate cells. This is seen in Cryptococcus, Saccharomyces cerevisiae, and other examples. Yeast-like fungi are the fungi which undergo incomplete budding, means the daughter bud does not get separate from the parent bud, so they keep on attaching themselves line by line. They form a structure known as pseudohyphae. This is seen in Candida and Trichospora. Molds. So mold has got true hyphae. True hyphae means they have got long, slender, filament-like structure. These filament-like structure can be of two types, so they can be septate or aseptate. In case of septate, the filamentous hyphae has got septa, means it has got transverse partitions in that hyphae, while in case of aseptate one, there are no such septa. And these true hyphae form a network like structure known as mycelium, which is further of two types. It can be vegetative, which lies on the culture, while it can be aerial, which lies above the culture. We'll be seeing what this culture is in our upcoming slides. Now lastly, the dimorphic form of fungi. It is the thermal dimorphism, means the fungi changes its form with temperature. So at 25 degrees Celsius it exists as mold, while at 37 degrees Celsius it exists as yeast. That means 25 degrees Celsius means outside the human body it will exist as mold, while inside the human body it will exist as yeast. Some of the characteristic example of dimorphic fungi include blastomyces, histoplasma, paracoccidioides, coccidioids, and sporothrix. These examples can be remembered by a mnemonic. Body heat probably changes shape. Right. Now, based upon spores, fungi can be divided into sexual sporing and asexual sporing. In sexual spores, we have got zygospores, ascospores, and basidiospores. In zygospores, zygomycetes produce zygospores, example Rhizopus. Ascospores is produced by Ascomycetes, example Aspergillus. And Basidiospores is produced by Basidiomycetes, example Cryptococcus. These asexual spores can be further of two types. They can be vegetative spores or aerial spores. Vegetative spores are of three types: arthrospores, blastospores, and chlamydospores, which are present in coccidioids, Candida and Candida albicans respectively. While aerial spores are of four types: macroconidia, microconidia, sporangiospores, and conidiospores. Macroconidia and microconidia are found in dermatophytes. Sporangiospores are produced by zygomycetes, while conidiospores are produced by aspergillus. Now, based upon vegetative state, it can be spiral, fabric chandler, nodular, pectinate, or racket type. Spiral looks like this. So you can see all these morphological structures, these vegetative state of the hyphae of various fungi. Now, spiral and fabric chandler are characteristically found in Trichophyton species, while nodular and pectinate types are found in Mycosporum species, and the racket type is found in Epidermophyton species. Now, based upon location, the fungi can be divided into superficial, subcutaneous, systemic, and opportunistic. Now, this location is the location of fungal infection which is caused by the fungi. So, it can be either superficial, like present over the skin. It can be subcutaneous, means infecting the subcutaneous tissue beneath the skin. It can be systemic, means all throughout the body, or it can be opportunistic, means in case of immunocompromised state, it manifests itself. So here we are just enumerating the examples of these four classifications. The superficial. In case of superficial, we've got Tinea vesicolor, dermatophytosis, Pedra, and Tinea nigra. In case of subcutaneous, we have got Mycetoma, Rhinosporidiosis, Chromoblastomycosis, and Sporotrichosis. In systemic, we have got all the dimorphic fungi except for Sporotrichosis, which causes subcutaneous infection. So in systemic one, we have got Blastomycosis, Histoplasmosis, Coccidioidomycosis, and Paracoccidioidomycosis. In opportunistic infection, Candidiasis, Cryptococcus, Aspergillosis, and Pneumocystosis. Now here we are just enumerating the example. In some other video, we'll be going into really great detail of each one of these infections, their causative organisms, and their clinical presentation in some other video. For now, just remember the examples. Now we come to the main part of this video, the lab diagnosis of mycosis. So fungal infection can have got really versatile clinical presentation. It can present with skin infection, it can present with nail infection, scalp infection, some pus formation, or some mucosal membrane in the mouth involvement, or any blisters form, or means any abscess, pyogenic infection, right? Now, depending upon the type of infection, the diagnostic method differs, because the first step of every diagnosis is the specimen collection. So it must be very clearly known by a doctor, or the one who is collecting the sample, that what sample should be collected and how it should be collected in order to proceed with the perfect diagnosis of the mycosis, right? So the steps of lab diagnosis include the sample collection and transport, followed by direct examination by microscopy and fungal stains, then fungus culture and identification and done, then serological tests, skin test, and lastly the molecular test. Starting with the sample collection and transport. So the sample collection and transport has got some really noting point. So the sample should be collected from the active lesion and not from the old burnout lesion. It should be collected under aseptic condition. The specimens should be of sufficient amount, and it should be properly labeled before being transported to the laboratory for the diagnosis. Now, the specimen depends upon the type of infection, if superficial, subcutaneous, and systemic. In case of superficial, which only involves the superficial tissues like skin, we can take the lesion scrapings. But one thing must remember, that scrapings are taken from the edges and not from the center, because in case of superficial, active lesion lies at the edges and not the center one. Then other ones are the nails and hairs. So these nails and hairs should be sent as a whole, right? In case of subcutaneous, pus, biopsy, swabs, scraping sample could be sent. For systemic one, CSF, blood, urine, sputum, pus, and biopsy can be sent. CSF and blood are more important as compared to the other. So okay, so this shows the skin scrapings for fungal infection. This is a good video which is demonstrating how to collect the sample, superficial sample from the lesion by skin scraping. So I'll be attaching the link of this video. Just go and check out it, right? Now second, the microscopy or the direct examination of the received fungi specimen, right? So in microscopy we perform two things, like wet mounts we create, or we use differential stains. In wet mounts we have got KOH Mount, lactophenol cotton blue mount, calcofluor white stain, India ink stain, and neutral red stain. While in case of differential stains, we have got a long list of stains like gram stain, HNE stain, Giemsa stain, PAS stain, or the periodic acid sieve stain, then Gomery methanamine silver stain, GMS stain, acridine orange, or Mayer's mucicormine. Now let's discuss some of these microscopic examination in a little detail. So KOH mount, as you can see, this black and white thing and these hyphal structures. In KOH mount we use KOH of various concentrations. KOH being a strong alkali dissolves the keratin which is present in skin, hair, or nail samples. So since skin contains less amount of keratin, so we use 10% of KOH for skin scrapings. For nails and hairs we use a stronger dilution of KOH, that is 20 to 40%. With the adding reagent we add glycerol 10%, which prevents the desiccation of the fungal cells, and we also add dimethyl sulfoxide, which digests tissue in case if the received specimen is a tissue specimen. Secondly, we have got this LPCB of the lactophenol cotton blue mount. So characteristically, we see blue structures under the microscope. LPCB is the most common stain which we use for microscopy for fungal isolates from the culture. Means the culture which we are creating for fungus, we take the fungi from the culture and create LPCB mount most commonly. Lactophenol cotton blue, so lactic acid which is present in LPCB preserves the morphology of this structure, and then phenol acts as a disinfectant. Cotton blue stains the fungi, and the glycerol which we add in addition to it prevents desiccation. Next is this calcofluor white stain. So this, as you can see, are fluorescence, right? So calcofluor white stain is a fluorescent stain which binds to the chitin fungal cell wall and gives fluorescence under UV light, right? So if we try to identify this fungi, we can say it's yeast fungi, right? Okay, it's not yeast like, it's yeast fungi because there's no pseudo hyphates. They're quite separate and they've got budding dot cells, right? Nextly, this India ink stain. This is a negative stain. As you can see, these refractile capsules over the dark background. This kind of a negative staining. We use it mostly for Cryptococcus neoformans. Next, the gram stain. All we know, all fungi are gram positive fungi, and we use it for yeast and yeast-like fungi, right? For and then H and E stain, where hematoxylin stains the hyphae and eosin stains the background. Next is the GMS stain, or the Gomery methanamine silver stain, which stains the fungi black while the background pale green. And important note here is that it stains both live and dead fungi, which is in contrast to the PAS stain, or the periodic acid sieve stain, which stains only live fungi. PAS stain stains the fungi magenta or deep pink, while the nuclei stain blue. Now next, following microscopy comes the fungus culture. So fungus culture can be of various types. They can be basal media, which includes SDA or the subarachnoid dextrose agar, neutral SDA plus some antibiotics, and the DTM or the dermatophyte test media. It can be nutrients deficient media, which includes corn meal agar, or rice starch agar. It can be enriched or selective media, which includes brain heart infusion or blood agar, or bird seed agar or Niger seed agar. Or lastly, it can be differential media, which includes the chrome agar. Let's discuss each one of these in a little great detail. Starting with the basal medium, the SDM. So this Severous dextrose agar contains 1% peptone and 4% dextrose. Its pH is 5.6, so it's slightly acidic. So it's inhibit the bacteria and it's ideal for the fungal growth. Now we keep it in a tube in a slant position. The slanted position is known as the obverse side, while this back position is known as the reverse side. This obverse side is used for observing the colony morphology, while this reverse side is used for looking for the pigmentation, if any, produced by the fungi growth. So as you can see, this is the obverse side of the SDA media. This is the reverse side of the SDA media, right? Now, since this SDA media can be contaminated, means any bacterial or other fungal growth can happen in SDA media. So we use some extra supplements into this SDA. So we add some antibiotics. So SDA, we add chloramphenicol or gentamicin in order to inhibit bacteria. Or we can also add actinidiol or cycloheximide, which inhibits the normal saprophytic fungi, right? Then other one is the neutral SDA. So neutral, as its name suggests, the pH is around neutral, so it's 7.2. Its constituents are 1% neopeptone and 2% dextrose. It is also known as the Emmons modification. Next basal media is the DTM, or the dermatophyte test media. It's nothing but the SDA, but we here add an indicator known as phenol red. So normally when it is uninoculated, it's orange in color, but when we inoculate it with dermatophyte, it turns red, because the dermatophyte fungi utilize the nitrogenous substances which are present in this medium, and they produce alkaline byproducts which turn the phenol red, means the indicator turns from orange to red, right? Now next, the nutrients deficient media. Firstly, we have got corn meal agar. So it's a media which is deficient in nutrition, so it creates an unfavorable condition for the fungi growth. In unfavorable condition, the fungi produce spores. So we use corn meal agar in order to produce spores, characteristically the clematospores. So this is how corn meal agar looks. It's quite transparent, translucent thing. Now one more thing, what we do in this corn meal agar is the Delmount's technique. So what we do is we create 4 streaks in which we inoculate the fungi from the specimen we have got, and then we put a cover slip over it. Now this cover slip covers these slits and create an anaerobic environment that stimulates the spore formation, and this modification is known as the Delmau plate culture modification. Right. Next, enriched and the selective media, we have got bird seed agar or the Niger seed agar, which looks like this, and the blood agar, as we all know, is characteristically red in color. Next, the differential media. The single one is the chrome agar, which is used for speciation of the Candida. The various species of Candida can be differentiated using chrome agar. So as you can see, the Candida species of various types produce various color colonies, so we can differentiate between the species of Candida. Now, culture conditions. So there are some optimum conditions which a culture must meet in order to grow the fungi to its optimum level. So temperature, generally we use a temperature of 25 to 30 degrees Celsius, but in case of dimorphic fungi, we have to grow the fungi in both 25 degrees Celsius as well as 37 degrees Celsius. Incubation time, generally most of the fungi take 2 to 3 weeks of incubation time in order to grow, while Candida and Aspergillus are quite fast growing, so they can grow within 24 to 72 hours. In antibiotics, as we have already discussed, we use chloramphenicol or gentamicin in order to inhibit bacterial growth, or actinidiol or cycloheximide in order to inhibit the saprophytic fungi growth. Right. Now, culture identification. So once we have grown the fungi over a culture, we have to identify it, like what fungi, what fungus it is, right? So this identification could be done microscopically or microscopically. In microscopic thing, we see for the rate of growth. So it can be rapid within 5 days, is seen in yeast or opportunistic causing fungi, while it can be slow, which takes around 1 to 4 weeks, and it can be systemic or subcutaneous, right? Then secondly, the colony culture, which is seen on the obverse side, the pigmentation, which is seen on the reverse side, the texture of the colony. So it can be black powdery as seen in Aspergillus niger, it can be yellow green powdery as seen in Aspergillus flavus, and it can be blue green powdery as seen in Aspergillus fumigatus, right? So as you can see, this black thing, this black powdery fungi is the Aspergillus niger, while this yellow green powdery fungus is the Aspergillus flavus. And lastly, we see the topography of the fungal colony, right? For the microscopic identification, we go for three methods: the Teased-Mount method, the slide culture method, and the cellophane tape method. The Teased-Mount method, we do nothing but we create an LPCP mount over the slide. We take the sample from the culture media and put it on a slide. We put some drops of the lactophenol cotton blue reagent, and then we put a cover slip over it, and is observed under the microscope for hyphaean spores. In case of slide culture, this has got an extra edge over Teased-Mount, but it's a quite tedious process. The slide culture is quite a tedious process. The extra edge is that it preserves the in situ morphology of fungal colony. Since in Teased-Mount we are taking the fungal sample from the culture using some probe or the loop, but in case of slide culture, we are not disturbing the colony. We are creating the LPCP mount over the petri dish only. So the morphology of the fungal colony remains intact, right? So as you can see, this is the slide culture. And the cellophane tape mount is also just like slide culture. It also preserves the in situ morphology of fungal colony, but it's quite easier process than slide culture. It's not tedious as slide culture. And in cellophane tape mount, we take the impression of the colony and we perform the LPCB mount. Right? Next, the serological tests. So in serological test, we can detect the antibody, we can detect the antigen, or we can do immunohistochemistry of the sample. Right? So in antibody detection, it can be done by ELISA, immunodiffusion method, agglutination reaction, complement fixation method. Well, these methods are discussed greatly in the chapter of antibody reactions, which will be discussed in some other video, so you can watch it. In case of antigen detection, we can detect cryptococcal capsule antigen by latex agglutination. We can detect galactomannan antigen for Aspergillus, or we can detect beta D glucan antigen for invasive fungi. Right? These antigen acts as the biomarker for the fungus in blood of the serum sample. Right? In case of immunohistochemistry, this we do for deep mycosis, where we try to identify antigen present over the cell, which are present, which are part of tissue specimen which we have received. Right. In case of skin test, this is used to demonstrate delayed hypersensitivity, used for histoplasma, blastomycin, and coccidioids. As you can see, this is how the skin test is performed, in which the small amount of fungus sample is injected into the skin, not very deep, and we try to look for some hypersensitivity reaction against those sample, right? Lastly, the most advanced form are the molecular test, which includes PCR, its variants like multiplex PCR, nested PCR, the real-time PCR, and the DNA sequencing. So in this video, we learned about the fungal structure. We learned about how to classify fungi on various criteria. We learned about the main thing, the diagnostic method of mycosis. Right? Now it's time for some viva voce. So what is the common stain for fungus isolates from the culture? What is the role of actidy on added to SDA? Which culture methods preserve the in situ morphology of fungal colony? What are some fungal biomarkers which can be detected in patient serum? And lastly, the question, all are dimorphic fungi except these are the options: Histoplasma, Sporothrix, Trichospora, and Coccidioids. Kindly comment your answers below so that I will reply whether you commented it right or wrong, right? So, like, share, and subscribe to this channel so that MindNotTired channel flourishes in future. Thank you for watching this video. See you there in the next video. Till then, keep your mind not tired.